I think the only way to start this off is very Monty Python-esque, and so now for something completely different, because it will be. I'm not a geneticist. I'm not a biometrician, and I don't work on wheat. So what the hell am I doing here? I work on crop ecophysiology and modelling, but I do work very closely with plant breeders, particularly in sorghum, which is the crop I work in most. And we've been thinking for a while about how to integrate that sort of biological insight that comes from understanding the dynamics of crop growth and development with the challenges faced by breeding. And you've already seen in a couple of the presentations the challenges that breeders face. And there's a whole lot of black box technologies in there that always, from my perspective, always sort of give me a bit of concern. I thought we should be able to do better than that. And so I was interested at the outset when Jessica put up her gain from selection equation and said, yeah, look, crop modelling might fit in over there in terms of selection accuracy and envirotyping, it's now called. We used to call it environment characterization. And so I thought, okay, it probably fits in there, but I think we can go beyond envirotyping. Okay, there's a lot where modelling fits that actually affects just about all of those terms, not just that one. We moved on to Matthew talking about physiological indices, but his conceptual model had this sort of nice quadrant thing, but was very static. He had in his head a dynamic model, but I don't think he's ever run a dynamic model. And so when you look at a number of the ideas he had in his static conceptual model, you sort of wonder where they came from and what would their relative impact be, and why would you choose one over the other? It's a little bit like the old physiology world where people said, this is a good idea and we'll do all this work and then the world will change. And so I don't know that we've connected well ideas with what their relative value might be. Then we heard a little bit about statistical models and genomic selection, and Jose, as always, does his nice sort of Greek alphabet, and that stuff's all looking really exciting, and there's clear advantages coming. But again, it's very black boxish, and you think, my God, can't we use this capability a hell of a lot better than we are now? And so they're the sort of things I've been thinking about. So I'm not going to bore you too much with detail. There's probably going to be a lot more in here about thinking about what to do rather than saying, here's what's been done. But I'll use some examples from sorghum work to try to highlight the ideas and what we're thinking, because it fits in with all those things. And to me, there's some amazing opportunities sitting out there to be grabbed, and by integrating these technologies, and we're just at the cusp of doing it, but we need to think a bit more about it. And so on the point of thinking, it's nice. I like to start with going out into the world of art. So this was a photo my wife took of me, actually, at the Museum of Old and New Art in Hobart, which is a really interesting, eclectic sort of place. And this was like a waterfall of genomic data just flying down a wall. And, it was just sort of rushing down. And so I thought, right, it just struck me that's where we're at, and it's not just genomic data, it's phenomic data. So you've got this amazing capability to collect data. And, thinking about the idea of predicting phenotype from genotype for complex traits, which is what I'm sort of into and most of us here are into, it just struck me that we're sort of lost in this waterfall of data. And to probably be a little bit bold, I'd say we're not really, though, we're starving for insight. And so this this is a parody of Rodin's Thinker that was in the sculpture garden in Des Moines, which I also thought was sort of a bit of a paradox, because the work we're doing with Pioneer is probably right in this space. But I thought it was interesting the artist used a rabbit, because it's sort of like the notion of fleetingness, so, not spending much time on the bits that are important. So we spend a lot of time, and it is important to get the data and get lots of it and get it well. But probably in general, I think we don't stop and think enough about what to do with it and what we might do differently if we can actually get data in different ways to what we have in the past. So we're still using old sort of style analysis technologies with new style data. It's starting to move, but we've got to get out of that and move on a bit. And that's where I want to go today. Think a little bit about that. So the bit that always strikes me, I always go back to this quote from when I first got into this stuff, which was like 20 years ago now by the look of it, talking to Cooper. And, this notion of adaptation landscapes was always one of interest. And so you sort of have this horrible complexity. Things float around with genetics, they float around with management, they float around with environment. And so you've got this sort of horrible adaptation landscape concept, which always, to me, just it grounds me a little bit in thinking about this, because you can look at this landscape as sort of an analogy of what you're doing. You're roaming around on this horribly complex landscape trying to find these peaks, and these might be different genotype and management combinations in one season or something. And then the season changes and this whole thing just sort of wobbles and becomes something totally different. And so the peak that you were trying to find up here suddenly becomes a trough and vice versa. And so you're sort of roaming around on this landscape. It's like crawling around a rugged landscape in the dark, trying to find a peak. And then the whole thing changes before you get there. So it's sort of amazing that plant breeders actually get anywhere. But they do. And those technologies, I sort of don't mean to be disparaging of black box technologies. They actually work. But are they the best way to do it, and can we do better? And the issue is about predicting and navigating. How do we predict? It's all about prediction, and I suppose that's what we've been talking about. And then how do we use that prediction to get us somewhere useful? So I want to cover that off in a few sections. So some conceptual sort of idea of where a bit of biological insight and modelling might fit. The sort of notion of coming at this from thinking about phenotypes rather than genotypes. So you'll probably be horrified at my concepts, but Matthew sort of raised this a little bit in terms of thinking about crossing scales of biological organisation. But from me being a sort of an organismal level biologist, I think about the organism as a whole and how it got to where it is, and then maybe coming backwards rather than starting with those arrows that sort of go up. The notion of how to introduce biological insight via crop models and trait dissection and phenotypic prediction, a couple of examples. I just realised, Lee asked me to introduce myself and I didn't even do that, sorry. So I'm Graeme Hammett from University of Queensland, for those of you who don't know me. And I also didn't go through that long list of people who have been involved in this work, so there's a whole group of people working on this. And the people working on wheat, Corinne is here and Scott was here, so they're the wheat people. If you want to ask any questions, you can ask them. But I'm also happy to entertain any questions on the way through, so feel free to just jump up. And at the end, I want to think about where we're going. So that's where I'll go. So the first bit is this sort of fairly sort of simplistic picture, but I think it sort of gives a good idea of how we might think about bringing physiology and modelling, integrating it with genetics and breeding. And of course we put the modelling in the middle. You know, it's like putting Australia in the middle at the start today. But of this sort of genotype to phenotype space. And what actually happens around this loops, and I think it's important to think of it as loops and not as things going one way or the other, because that's really how it works. And so if we wander around this, you can sort of think, if you've got some dynamic framework that captures insights about growth and development of crops in a dynamic algorithm of some sort, then you can start to predict what might happen if you change things, evaluating traits in TPE, target population of environments, and environment characterization. So, and I know Corinne's going to talk about environment characterization, so I'm not going to talk much about that, but it's a key component. And if you've got a phenotype, then you can dissect it. And if you understand how complex traits, so how do you get to the phenotype from some component, interaction of components? And a modelling framework actually helps you understand and dissect that out. It helps you then detect the genetic architecture underpinning that, because it helps you think about how to phenotype for it. And then you can connect back by looking at how do those regions that control those underpinning causal factors associate back into the model, which enables you to predict, and you can keep going around. So that's the sort of logic flow of what we do. And I'll come back to some examples around that. But the notion is to try and really bring some biological insight into the process, not just because that's a good idea, but because hopefully it'll make a hell of a difference in that genetic gain equation. And I think it can. So phenotypic distance, the second bit about this, and as I said, Matthew introduced this before, so my diagram's a bit clunkier. But essentially you've sort of got this notion of scales of biological organisation, okay, where we're often looking at genome level information and some sort of phenotype out at the crop scale, but there are all these sort of levels of organisation in the organism that you have to jump through to get from one end of that to the other. And it was even the genomic prediction stuff now is still lost in the paradigm of jumping from here to here, okay? How do we predict yield if we've got a whole lot of data on genomic profiles of a lot of individuals? So it might be better than some of these other things that we've used in the past, but it really is just another way of tweaking this to get a prediction from one end of the spectrum to the other. Is that necessarily the best way to do it? And the problem that still is confronted by is this notion of context dependencies. So you've got, you do a training set and you do a prediction and you run it off into another environment and the thing just goes over a cliff. And so you haven't addressed the core issue of because you're still using linear models of what's a genetic background effect, what's an environment context effect. So all of these procedures are still constrained by those context dependencies. Now that doesn't mean any of these other things can do any better, because you can sort of start at this from bottom up, and there's people who look at sort of gene network models and try to say, well, I understand every pathway, every biochemical reaction in the flowering pathway or the photosynthesis pathway, so I can make a model that will go enzyme level up, and you get something like iPlant or something like that, which it's sort of interesting but makes a lot of absurd claims about being able to model what an organism will do by pursuing the dynamics of every enzyme in it, and we're just nowhere near that. But you might be able to do something useful to get you a bit of understanding. But at the other end of this, the area where I tend to sit is more in these whole plant models, where we sort of come down from the phenotype and look at how organs and plants and things organise themselves and how that interacts with genetic architecture and environment. But it still only comes down to sort of somewhere around the organ level. So you're not able to span this. I'm talking about complex traits, okay? So I like the idea of thinking about phenotypic distance. So, and I think some of you were just sort of talking about this before when you started discussing some quality and other attributes. So there are some things that scale sort of directly or fairly directly from that sort of molecular genetics level to a phenotype. And some of these things are the sort of more, probably more higher heritable traits, but things that's, and a large part of the reasoning for that is because they do scale directly. You haven't got a whole lot of intermediate pathways you've got to go through. You've got something that expresses something that gives you the phenotype. And that can, quality, disease resistance, a lot of those traits, salinity, tolerance, they're all sort of, they're complex in their own right, but they're not complex traits. And so you've got this sort of, what I call a short phenotypic distance. There's probably words that geneticists use, but to me, that's what it's about. And so in those sort of instances where you've got few context dependencies, you don't get too much complexity, then you can start to make better use of these sort of gene network models or these sort of direct genetic prediction models. But when you move into a longer phenotypic distance where you haven't got that, where somehow, whatever happens down here has to go through a lot of interactions in the organism before something gets expressed out here that you measure, then, the whole thing's confounded. And so you've got this context dependencies on environment and on genetic background. And that's where this notion of looking at connecting capabilities that we have at the understanding of crop growth and development and a quantitative understanding of it might help in linking to things like genetic prediction and quantitative genetics models. So instead of trying to jump from here up to here, we can break it down a little bit and make the thing targeted at things that are probably much more tractable. And I think that's on the way, and I'll get towards some examples. But we have to think a bit differently about how to do things to do that. So the third bit I want to talk about was just a little bit about crop modelling, so I won't bore you for too long. But to introduce biological insight into this process, you actually have to have a bit of an idea about how crop models work, and I can show you it's not in Greek, mostly. So the simple view of this is just sort of, box and arrow thing where we can break a crop growth down into the development component and the growth component. And this is just running the life cycle of the crop through its developmental stages. So this is any determinant species, cereal, for example, wheat, sorghum, whatever, maize, and the growth side of it. So, we can look at how this thing progresses through its life cycle, how that's controlled by temperature, photoperiod, water, nitrogen, how that actually generates how many leaves or tillers that plant and crop are going to have. That then interacts with this growth component, which is really, how do we get to grain yield? We need to know grain number, we need to know grain size, we need to know the growth rate, and particularly around anthesis, which drives these things. And that growth rate is driven by either capture of water or capture of light, and the efficiency with which light is used or the efficiency with which water is used. That depends on where the roots are, depends on how the roots work in relation to the soil water. The radiation interception depends on how what the leaf area of the canopy is, what its architecture is. That depends on leaf number, which comes from over here, depends on temperature, depends on a number of environmental things. This depends on nitrogen. So all these things sort of interact, but there's a lot of this stuff we know about. You know, there's certainly a lot of subtlety in how one genotype varies from another that we don't know about, but certainly a lot of the basics we do know, and essentially this is what a lot of you guys see as G by E. And in a crop model sense, it's admirably explainable, okay? And so the problem is the crop model looks more like that, and I won't wander through this in too much detail, but all of those sort of simple boxes and arrows are actually contained in a whole range of interconnected equations that capture all of those biological interactions, okay? So, for example, the phenology part, you see here flowering, leaf number. There's all these environmental inputs that drive this sort of thing: radiation, temperature, photoperiod, vapour pressure, rainfall, soil, water. So you've then got some notion of canopy development. How does the canopy grow over time? These green boxes here actually connect through to a nitrogen sheet like this, which is equally as spaghetti looking. And here's the piece on growth rate. So light use efficiency, water capture, crop growth rate. So you suddenly start to use that canopy to intercept light, capture resources, grow the canopy and grow yield over time. And so all a crop model does is put that into equations that interact with each other. So the interactions that emerge out of this are, as I said, what you often see as G by E. And so a lot of our work is targeted at getting those equations representing the biology well so that we can do predictions that capture those interactions rather than just do them with linear statistical models, because this is non-linear, it interacts with the environment, and so it's not going to be captured by a linear statistical model.
Question.
Yeah.
What of those do you measure, actually?
I'll show you some experiments in a minute. But we, you know, in these sorts of, we will set up experiments specifically targeted at one of these arrows, okay, to understand what's going on. So we might measure light interception and biomass accumulation. We might measure water extraction and biomass again. But we won't, you know, you're not going to do that on thousands of lines. But you can, if you do that on a small number of lines, you can understand how it works and then think about how to phenotype the thousands of lines. So the experiments that we do to get the understanding are very different from what you would measure in a plant breeding context. I'll come back to it. If I haven't answered the question, ask it again. So we've put all this into a platform, AppSim, the Agricultural Production System Simulator, which has been wandering on now for over 20 years, but it has a number of purposes, and it was set up initially to run management of crops and cropping systems, and in my group we've really adapted it to start looking at genetics as well. So the thing really runs a crop or a cropping system through time. You set up a soil, you set up a weather input, you set up management inputs, you tell it what crop you want to run. So actually you are running a field through time, and you give it all the inputs as a crop would get, and it will give you the outputs of the crop growing and developing over time. Often on a daily basis, or on some of these things a hourly basis or less. But often these things run on a daily time step. So what we've done is rather than, a lot of these crops have a lot of generic processes going on. I mean, photosynthesis is photosynthesis and root growth is root growth. So you don't really have to have them different for every species you play with. And so you can do a lot of generic mathematics across this. And so you connect the processes up to get this crop growth and development in a fairly generic way, and then you can start thinking about how to link that with what you understand about genetic architecture of particular things. And how you get to understand that, I'll come back to. But again, it's a little bit about perhaps thinking a little bit differently than the conventional wisdom. So just to show you that it actually, what the level of prediction is, and these sort of things do work reasonably well. This is our sorghum model validation. So, people often put these things up and say, oh, here's the observed, predicted, look, it works. What we do with this is we have a lot of data, we have a lot more data than this. But I tend to only want to use the data to do this where I know if the model doesn't predict the data, it's not because of the data. And so these are data sets that we have what I would call extremely high confidence in their quality. And that also means soil, environment and crop, not just yield of the crop. I mean, you've got to have detailed measurements of a whole range of things. And each one of these points, we can put soil water, soil nitrogen or whatever under it. So these are a range of genotypes, soil water conditions, soil nitrogen conditions, different continents. So there's a fair bit of work gone into getting this thing as robust as it is, and there's crop models and crop models. It's a little bit hard to say it, but you can get a picture that would look like this by changing coefficients for every point you've got there. Okay, you can get it to fit, but to get it to fit without changing the coefficients across a whole range of conditions is a little bit harder, and means you're testing the biological insight inside the model, and that's what we've done here. Yeah.
It looked like your predictions for biomass were more accurate than your predictions for yield. Why is that?
Yeah, well, there's a bit of an over-prediction of grain number in this very high-yielding stuff. So there's a bit of bias at the top end here. We're not... That's where we're at. Yeah, I'd say we're working on that grain number bit because it works, the algorithm that it works off, we'd probably like to improve. But, yep, that's where it's at. So what it's telling us is that we don't understand that grain set as well as we'd like to, particularly at those high grain number situations. So this comes back to the experimentation. So in doing experiments for this sort of stuff, we look at how do we quantify control of the dynamics. So the thing I mentioned earlier is that, like in Matthew's talk, it's sort of okay to think about what trait do you want or what index do you want, but often what's really causing the consequential phenotype is driven by dynamics through time. And so you really can't capture that in a simple index at a point in time very well. So the whole thing is about dynamics. And so we run experiments where we will target specific arrows in that spaghetti diagram so we can figure them out. And so here's an example with different height sorghums looking at light interception and biomass, looking at light use efficiency, for example. So, differences in light use efficiency across different genotypes. We look at platforms to measure transpiration efficiency, so we can quantify how efficiently different genotypes convert water into biomass. And so we set up systems just to look at that bit. Tillering, so field studies looking at control of tillering over time. Again, a sort of a very important point in the development of canopy, at least in sorghum, but I'm sure also in wheat. Water extraction, so we do silly experiments like putting things under a rain-out shelter with sorghum rows 6 metres apart, plugging them with neutron probes, just to see how a root system works. How effective is a root system at capturing water? Where can it get it from, and how much can it get? So you get these sort of graphs of water extraction and spatial water extraction. So that enables you to sort of inform a model. A lot of work on roots that I'll come to, but we then pursue that into some of the genetic architecture of root systems in more controlled experiments. So the answer is there's a lot of detailed stuff, and that was the reason I asked Matthew the question about phenotyping things off to the side of the field. There's a lot of field-based stuff, but there's a lot of stuff. Once we've got a lead on something, you can measure it in experiments with plants in funny boxes like that, and I'll come back to the fact that that was actually a very useful experiment. But the overarching notion is that one, to sort of, connect the physiology to genetics, really, by looking at the coefficients that enable you to predict the dynamics, and get some idea on how to phenotype it, and also then enable you to do this sort of simulation, predicting what's going on. So, as I said, just break in wherever you like. This is going to be sort of a bit different from the stuff you're probably used to, so feel free to stop me. The fourth part of this, I wanted to just cover a few examples. So we've done work on a range of examples, and I just want to pick two of them. So in looking at dissecting traits and then predicting what a phenotype will do, Corinne, who's here, did a lot of work with her postdoc on maize. That was really nice, and was probably the first example of taking things right through from QTL to effect on crop yield. Some really nice papers. I'll give you the example of the work we've been doing on stay green. There's work on tillering. There's work on root system architecture. It's a little bit of that I'll pick up in the stay green example. And some work on maize on this sort of maximum transpiration rate trait that affects canopy temperature, and some recent publication on that. So I'll just do those two examples to show what we're up to. And stay green, I suppose people working in wheat know about stay green. So the idea here is to look at this part of the loop, the dissecting a complex trait and detecting its genetic architecture. So that's the bit we're looking at. So stay green we can see here. In sorghum, this is a stay green genotype versus a senescent one. All the leaves are dead, and this will yield more under drought conditions and will have reduced lodging. So it's a fairly important trait in our environments. And in the past has been screened just visually in breeding trials if and when it occurs. So David Jordan did a nice analysis of this from all his breeding trial data, looking at the effect of stay green on yield. He had a number of male parents crossed with many females varying in stay green, and then he plotted those and looked at the sort of yield stay green score association. So the next plot shows you the slope of those thousands of, well, not quite thousands, but hundreds of graphs. So this is the mean site yield, and this is the slope of that yield change per unit of stay green. And so each one of these is one of those populations. So here you've got high yield change per unit of stay green, and at yields below about 5 tonnes per hectare, you've got generally positive effects. Up here, you've got not much. It's a bit all over the place. Some significant ones, but significantly negative as well as significantly positive. And so, as I put here, mostly we're operating down here, but there are a number of locations and environments where we do get yields of this order. So you can see that immediately this sort of effect of the environment can overwhelm this. So the genetic regulation of this is quite complex. So Emma Maize done some nice work on this, and Andy Borrell, and they've mapped and looked at all of the published stay-green QTL, and you can see there's a large number of small effect QTL, but there are 4 major ones that people have been playing with, and they generated a set of near isogenic lines to look in more detail at those.
I have a question about how you phenotype the stay green. How do you distinguish that from differences in days to maturity? It's done at a standard time, so they'll know the field phenotyping is just a visual score, so they'll just walk through and give it a number between whatever 1 and 10 of how stay green it is. But they will do that, and they know the flowering time, so it can be adjusted for that. I think they try to do it at a standard time, or at least that can be adjusted for. But it's very much a breeder's trait. They just walk the fields and score it. So when we looked at what might cause stay green, and Eric Van Oostrom and Andy Borrell were the people really leading the physiology piece, looking at the near isogenic lines and also contrasting hybrids, then they sort of came up with this picture, which is, in a way, a sort of a simple model of it, the things that could affect it. So ultimately, we were hypothesising that this was really about changing the water balance around, and that you could possibly change the water balance around by changing root architecture, which might increase your water accessibility, or you could change the canopy through tillering or leaf size or flowering, which would reduce water use up to anthesis, enhance the water availability, or you could change the transpiration efficiency, give you increased growth rate, give you increased yield. So there are a number of ways of changing these sort of things that would flow through this sort of dynamic to give you a stay green phenotype, potentially give you a stay green phenotype and increased yield under drought. And so these were sort of the traits that we went and looked at. And so just to look at tillering, for example, so and these make nice studies for PhD students. So these sort of studies mostly come out of postgraduate work. So reduced tillering. So I'll just quickly go through these. So one of the things that we could see, reduced tillering is associated with stay green, and you could get these co-location of QTLs for tillering with some of the stay green QTLs. And so Andy Borrell's published this recently. So here's your green leaf area, I think it's green leaf area at flowering, and culms per metre squared, and these are the different stay green. This is the senescent, and these are the introgressions of those QTLs. And so, one of the things you get is just reduced tillering. Okay, so it sort of seems obvious. You just have less rapid growth of a canopy, saves itself a bit of water, but that lines up pretty nicely with the genetics. Another one of these is to do with root architecture. So you can look at water capture at depth, and we did a number of studies on root systems and determined some of these QTLs associated with root angle in sorghum, and that also lined up with stay green QTL. So here's some of the pictures. These are
In sorghum, you've got to really wait and look at the nodal roots rather than the seminal roots, because they frame the main root system architecture. These boxes are 1.2 metres deep and 1.2 metres wide. So these are sort of not studies you do in a hurry. But this was after looking at parental lines from the breeding population, identifying some variability, and then looking at specific lines from across where we had very few differences in above ground growth, but known differences in the root angle of the nodal roots below ground. And these are the sorts of different looking root systems you get, and you can immediately see this sort of, this is a bit of an extreme picture, I should add, but you know, there was a common feature through these of, you know, less occupancy of roots at depth under this wider angle plant. Single plants in boxes, so you know, it's still a little bit unreal. But the nice thing you can do with this, and this is where it connects back to the breeding really well, is, you know, we can then phenotype root architecture in these sort of systems, and we can do it in small containers, which we worked out a way to do this, that you can phenotype whole populations. And we do that for the parental lines, and we're now doing it for some of the NAM populations. And so David Jordan's got this really nice setup with various mapping populations, NAM populations, and a lot of these things, you know, multi-environment trials with multiple years of yield data. So we can then identify regions, project them onto the consensus map, and then look back at how does that relate to the performance data in the field. So it's a sort of validation, in a sense, using the database from the breeding program and through the genetics, rather than us trying to run experiments with things that differ in root systems and trying to measure soil water and whatnot. And of course you can then start going down to candidate genes once you've, and with the NAM populations that you get to this, with the numbers that we can now do, you can get to that. So the point with that was that we mapped this initially in a fairly simple study where we just grew the plants in between glass sheets and did one of the populations to identify regions that controlled root angle. And it was pleasing to see that they didn't co-locate with anything that related to above ground growth. So they were independent of things going on above the ground. And then you can map this back onto David's breeding populations through those regions. And you know, he's then got a number of real hybrid combinations where we could look at these QTLs from across those populations from field data and just test the association of that region with yield. And so, you know, there was an association between that region and effects on yield, and it was to do with the allele associated with narrow root angle. So, you know, we can fairly quickly identify a phenotypic measure that's a sort of morphological thing that connects to soil water, does all the things it needs to do, but then can also, we can validate it back through the genetics program database. So we're now phenotyping furiously for that with these NAMs. So the last one is TE. So you can look at transpiration efficiency and there's a number of things that can affect transpiration efficiency, and one of those is to do with this limited maximum transpiration rate, which is what I was asking Matthew about, because this mechanism actually will give you a high canopy temperature, because what it does is you slow down the water use in the middle of the day under high vapour pressure. So that's the time of day a plant is using its water least efficiently. So it's a good time of day to close down, provided you don't get too hot, but you will get hot. So in that instance, you want a plant that will get hot in the middle of the day, not one that will be cool. for one with more effective root systems, you probably want the one that will be cool. So if you're just phenotyping for canopy temperature and you don't know which one you're looking for, or you're not, you don't know about the environment you're in and which one you're getting, you might be throwing away the wrong stuff, okay? So you need to be aware of what the indicator is actually doing for you. And in this instance, if we looked at some of these stay green 1 introgressions, under the higher VPD environment, it tended to have a higher transpiration efficiency. So that's suggestive of this mechanism where this thing's slowing down its water use under these high demand conditions.
Sorry.
Yep.
But in which situation would you want, for example, if you're in the field and the plant in the canopy is warmer, usually this is correlated with a lower yield.
No, in this case not. In this case, the warmer one will have the drought adaptation. That's what I was saying, you've got to be careful what mechanism you relate things to. And the same thing happens in wheat. This trait's also been discovered in wheat. So the stuff that Matthew was talking about is fine. Where you're looking at deep roots in a deep soil and you get a nice association of yield with cool canopy temperatures, that could be an index for selecting for narrow roots or for a good root system. It could also be an index for selecting for poor transpiration efficiency. So you've got to be careful that you're not selecting for the wrong thing.
Sorry, Graeme. Just to clarify for you, an easier way to think, well, I think Graeme probably already said this, but that's if you're looking at them under irrigated conditions, for example, you might find the one that's heating itself up, so its roots are clearly not under stress. So, and under those conditions, it may well not be the best yield. In fact, probably isn't. But if you then grew it under drought, it would still be higher for some time, but eventually when everybody started running out of water, it would be cooler. But you might not see that at the time. So eventually it would probably be cooler than the guy that fails, because that guy will run out of water first.
Yep, there's a
So
temporal
there will be a switch during the season when you will see, yes, you'll see the correlations you guys see, but it will switch. So if you looked at them early, you chuck out the wrong ones. If you looked at them under drought,
But if you follow, for example, the temperature along, for example, the crop
the
cycle,
whole time.
so you will see, eventually you will see.
Yeah.
this
You should see the crossover, yeah.
But this would let you select an irrigated environment for drought adaptation, which is what's really useful to do.
And that's the point, I think, is just knowing what you're looking for and how to design the system to find it and not confound it when you've got things that give you contrary, the same measure can give you contrary mechanisms. So to move on to the prediction part of this, so coming back around this picture, how do you then connect that back to a prediction of the phenotype? So you can sort of use the model to understand the genetic variation, but you can also think about connecting it back together to predict what it might mean for you. And that's where you come back to the spaghetti diagram. Scott's here, so I can blame Scott for this picture. And so here you can see, all I've done is just highlight. You can introduce some changes into this picture that actually change the traits I was just talking about. Okay, so you can say, what if the tillering, we change the tillering a little bit? What it'll do is it'll change this whole leaf area dynamic. What if I change the TE a bit? It'll change this relationship with VPD. What if I change the roots a bit? It'll change this water extraction. And that's all you change. The rest is just the model wrapping itself around that change and all these interactions that then occur with that change and with the environment you're growing it in. So you're not telling it anything about yield. You're just telling it, I'm giving you an extra tiller or one less. I'm changing your TE a bit. I'm changing your roots a bit. Now you wrap yourself around that and see what comes out the bottom when I give you a certain environment. And when you do that, this is the example. If we go back to stay green, low tillering, and limited transpiration. So these pictures are what we would pump out of an AppSim simulation, where we'll look at the trait variation and simulate the dynamics. And this side is the plant available water content. So this is soil water, and this side is leaf area. And I'm going to read this. The low tillering one is the red one. So you can see, this is a particular season I've picked out to make the point. But this is a terminal stress. So flowering's about here somewhere. And, these two lines hardly look different, okay? You've got a slightly lower tillering line, the red one, that gives you a little bit less water use. It might only be 5 millimetres or 10 at the most. But that amount of water gives you this difference in leaf area. So it's hardly a measurable amount of water that you're talking about here. If you try to measure this with a neutron probe, you probably wouldn't find it. But what it's saying is, if you have that slower water use, you will generate this type of leaf area pattern. Your leaf area is sustained a bit longer, and you've got lesser leaf area because you've got lesser tillering. So your leaf area is smaller, but then that little bit of water you save enables you to stay green longer. So you haven't told the model anything about stay green. You've told it, give me a slightly less amount of tillering, and you've generated a stay green phenotype. So that to me is a nice test of the biology in the model. And similarly, when you do limited transpiration, the same deal. If you restrict the transpiration under high VPD in the middle of the day, doesn't use the water quite as rapidly, has the same leaf area capacity because you've got the same tillering, but it saves that bit of water and can stay green longer. So you can, in these environments where you've got a severe terminal stress, the ones that normally highlight the expression of stay green, we can simulate that.
Yeah. Back to that last slide, does that then give you the ability to test one version of stay green against the other as to which will perform better in a crop?
Sure does. So you can start looking at mechanisms, or you can look at, whether these mechanisms are independent genetically is another question. Yeah, they may not be. But you can then look at that. That's a particular environment to highlight the point. If you look across seasons and you just take a low tillering type or a limited transpiration type and you look at the yield difference against the yield, so this is the low tillering type minus the standard, you can see there's a lot of years here where you're on the wrong side of the axis, okay? So this is the environment interaction and the it depends thing. So when you get up to these yields above 5 or 6 tonnes, if you've got a low tillering type, you're losing a lot of yield potential. You're making good gains down here, but losing it up here. And the same with a limited transpiration one, but to a lesser extent. And that probably reflects this picture from the field data, where the effect's much more obvious at these yields below 5 tonnes or so, and much next to nothing at higher yields. So you've got these trade-offs going on, and these G by E sort of things. And so you can actually simulate those as well. So what does that mean in terms of phenotyping? And I'm not going to go too far into all that, but what we have got into a bit, I suppose, is if we understand what's going on in these sort of traits and how they are perhaps more causal of the consequential phenotypes we're seeing, then what we want to understand is what's controlling these traits rather than directly the phenotype. And so we then do things like the remote sensing with UAVs that Scott's been helping us with, doing image analysis. So here's an image of a sorghum plot and then an image analysis to
try and do head counts so you can start to get tiller number. We can do targeted phenotyping systems of root angle and run whole NAM populations through them. And we can look at transpiration efficiency in some detail, and we're just developing a more higher throughput system with smaller pots of this. So the idea is to shorten that phenotypic distance. So we sort of, instead of saying let's look at the phenotype at the end and have all these horrible genetic and environmental context dependencies, we can probably get rid of them, get rid of a lot of that if we look at these things and let the model deal with those context dependencies, because it can.
If you are interested in one trait,
Sorry?
okay, if you are interested in one trait, this has a phenotypic distance that you cannot change.
That's right. But what we're interested in this trait for is not the trait, well, we are interested in the trait per se, but the genetic architecture of that trait will flow through to a whole lot of consequences on yield by interacting with the genetic architecture of these other traits. Yeah. And so, you know,
how do you shorten the phenotypic distance?
Because we measured this rather than yield. So, this is in fact far more highly heritable than yield. Okay?
You select for this, not for yield.
Yes. Yeah, and then we model yield. Or we look at modelling. We can take the variability of those things out with a model. And we can combine them. Okay.
So the idea is also that these traits are more simply inherited.
Yep. Yeah, and you might target your genomic prediction at these things. And then, by putting that into a model, you can then generate basically epistasis and G by E.
So you took only positively correlated traits.
Pardon?
You took only positively correlated traits.
For 3 correlated traits.
No, 3 correlated traits.
Sorry, I'm just not understanding.
Positively correlated traits.
Positively correlated traits. Positively correlated with what, sorry?
Yield.
For example, yield and tillering.
Okay, yeah, none of those things are positively correlated with yield. If we go back one, here's your yield consequence from changing some of those traits. So it depends totally on the environment you're in. So they're not directly, there's a lot of interactions beyond those traits that get you to yield. And so there will be associations of these traits with yield in some environments, but not others. And in fact, in others it would be negative. So it's about extracting your understanding of these traits to help you understand what you see in your phenotypic data at a yield scale. But yeah, they're not simply correlated to yield. The other example I wanted to give, if I've got time, I'll just keep going, was about the work we've been doing with the people at Pioneer on limited maximum transpiration efficiency in maize. And so the idea here is to, we did some work in sorghum, some sort of hypothetical work where we, 5 minutes, where we just looked at changing TE and what it meant for harvest index. And so, we got this sort of thing going where we look at this yield as a resource capture, resource use efficiency partitioning sort of model. And if you look at, this is vapor pressure deficit, that's the dotted line, that's what happens during a day. So in the afternoon, your vapor pressure deficit is high, your transpiration rate normally follows that, then comes down because the lights go off before the VPD comes down. In terms of the sun setting. So if you can look at limiting the hydraulic conductance of a plant by putting a limit on the flux through the plant. And if you do that, then you get this sort of thing where, again, you simulate no restriction, the open circles, or restricted water use, which will be hotter, okay? By the time you get down to here, it'll be hotter. But it saves some water and can use more of it during seed filling. And you get that same sort of picture. So you get this sort of picture where when you look at relative yield against yield under these at a range of locations, so these are all simulated data, up to yield levels of 4 to 5 tonnes, you tend to get positive effects from this sort of trait, whereas you lose a bit under the high yield conditions, which is understandable because if you slow down the water use, you're closing the stomata a bit, so you're also limiting CO2 assimilation. When you've got plenty of water, what you want is those stomata open and making most use of the light. So again, it depends which end of this picture you really want to position yourself at. So same thing happens in, these are the sorghum, so you can phenotype for this looking at transpiration rate against VPD. You get sort of genetic variation where there's different levels of cut-off for this. Some genotypes just keep going, others slow down under high VPD. Same deal in maize. We can do the same simulation in maize where we look at photosynthesis during the day. We can look at what that means in terms of transpiration during the day. We can impose this either something that keeps going or something that will slow down. So you get this sort of effect where one type slows its water use down in the middle of the day. And when you put that onto corn in the US, Charlie's just published this, and they've got a nice system where they can simulate the whole corn belt. So this is the US corn belt, and these are yields, fortunately, in grams per metre squared. So, you've got very high yielding crops in this northeast through to sort of lower yield. So this is the sort of average yield simulated over whatever it was, 50 years in the corn belt. But what you can then do is say, well, let's look at what happens if I put that trait on. And this is this is yield difference, okay? So this is about a tonne positive, this is about a tonne negative. So if you put a limited transpiration rate on maize and run, this is the sort of 50-50 point. If we run, 50, 100-year simulation, these are the average sort of the 50-50 point yields. And so for a lot of the western part of the Corn Belt and a lot of the Midwest, there's not much effect through here, a slight positive, big positive over here, some cost to that over here. But when you do it in the best 25% of years, so these will be the high yielding years, you lose quite a bit in these high yielding parts, you're still gaining over here. But in the worst 25% of years, the sort of, the lower yielding years, you get a significant advantage everywhere. And so that's the sort of simulation game you can play. How much of it you believe is another matter, but at least it gives you an indication of what's likely to be the case given what we know and how it hangs together in a crop model. So that's the sort of stuff, and we're starting to look at doing that for sorghum in Africa and India, for example. So where to from here, just to finish off in my last few minutes? I sort of like to think about how to integrate these new technologies and thinking a little bit differently about how to do that rather than just accepting the way we do things now. And the notion of serious play with models, which is what we do, sort of helps us think about that. And the work we've been doing with the guys at Pioneer is really interesting because they sort of have this idea of running around this natural system, mathematical system, and doing simulations and also connecting that with genomic prediction possibilities. But we can now actually simulate those adaptation landscapes and look at breeding trajectories on them. So I'm not going to go into the detail of this, but you can start looking at multiple traits and breeding selection strategies and run these things through and see where a breeding strategy might take you. So you can start to do experiments on breeding systems. But the bit I think we've been talking about today is more about how do you enhance genetic gain by adding more power to selection. And I pinched this picture from this Advanced Research Projects Agency, US DOE thing and I know a number of people I think here are involved in these projects, and it was really good to see a big bucket of money being put at how to integrate technologies, even if it was targeting sweet sorghum or something that I thought was a bit strange. But the idea of integrating all those technologies and how to go about it and actually having some projects trying to do it is really good. The one major shortcoming I see with their picture is the fact that they've only got arrows going one way. And really, I think the power of doing this is actually having the people doing the breeding in the centres or wherever actually having a far bigger effect the other direction than all this being, I will do all this and the world will change. So, I think that'll happen anyway. So just to finish, I want to come back to this notion of phenotypic distance, about complex traits not scaling directly, and how might we actually do something in that space. And I think there's a few things that can be done. One is about thinking totally differently about phenotyping. And it might just be, for example, I've got it down here, model-assisted phenotyping in training sets. There are lots of things, now that you can do genomic prediction and you've got training sets and bigger genotype sets and different environments, you can structure environments, you can think about phenotyping in a very different way than you have before, and you can start to use models in helping you do that. Certainly trait discovery and trait genetics, it's a sort of a no-brainer. And this notion of trait by E complexity, I think if we start thinking about how to integrate some of the capacities we can do with quantitative modelling, with what's happening with the statistical world, then you're going to be able to do a lot more than we're doing now. And it's not about environmental covariates, it's a lot more than that. You can start to extract what you know. So with David Jordan, we're looking at ways of running his breeding trials, simulating each plot, extracting the effects of maturity and tillering, and doing genomic predictions on what's left. And that to me gives you far more power in terms of saying, well, I know these bits will give me background effects and G by E effects, so I can model them out. So let's take them out and then look at what's left and do a genomic prediction on that. And this paper, I don't know how many of you have seen it, it's just come out, again from the group at Pioneer, who have in fact put a crop model into a Bayesian computation method, and they've actually got this working basically. It's all in virtual world, but essentially they're saying, they can optimise coefficients through a simple crop model and outdo G-BLUP by doing this with what they call genomic crop prediction or something. So they put a crop model into a genomic analysis context, and they can, particularly when they get outside their training setting into other environments, they demonstrated how it outdoes G-BLUP. So I think that's where we can go, and it's starting to go there. But I think we need to look at all the technologies and try and figure out how to integrate them far more effectively. We get funding from a whole lot of people to do various bits of this, so I'd like to acknowledge them. Sorry I took so long, but I'm finished.